Assalamualaikum warahmatullahi wabarakatuh. This is a presentation for the self-assembly properties, specifically on the application for the molecular Lego, presented by me, Nur Syafiqah binti Ahmad Roswai, as a coursework for the subject Bio-Nanotechnology. So, for the introduction, I will talk about the self-assembly. So, what is self-assembly? Self-assembly refers to the spontaneous molecular arrangement of the disordered entities of molecules into ordered structures, which is resulting from the specific local interactions among the components themselves. Simply put, self-assembly refers to the aggregation of particles into ordered structure without applying any external assistance on the particles. So now, this is the examples for the self-assembly properties in the biological material. The first one is the construction of the cell membrane. As we know, cell membrane consists of the phospholipid bilayer. So this is the figure depicted for the construction of the cell membrane. The second one is the helical structure of DNA, which is refers to this figure. And the third one is the folding of polypeptide chain or the protein chain, which is depicted in this figure here. So, these are some of the examples for the self-assembly properties in the biological materials. So now, these are some of the interactions or the forces that driven the self-assembly properties. So the first one is the van der Waals forces. The second one is the hydrophobic electrostatic force. The third one is the hydrogen bond. And the fourth one is the pi-pi aromatic stacking. And the last one is the metal coordination force. So now, this is one of the application of self-assembly properties, which is the molecular Lego. So what is molecular Lego? Molecular Lego, also known as the type 1 peptides, which can form beta sheet structures in the aqueous solution because it contains two distinct surfaces, which is the hydrophilic and the hydrophobic part. Why is it called as the Lego? Okay, as the Lego that we placed before, which have the structures of pegs and holes that after we assemble it, it can form into particular structures. These peptides can do so at the molecular level. Now, I will talk about the application of the molecular Lego from a research paper that I have found, which is photosensitizer encapsulation for photodynamic therapy or PDT. So first, what is photodynamic therapy? PDT. Photodynamic therapy is a kind of treatment that uses a drug which is called as the photosensitizer or photosensitizing agent, which is a type of light. So when this drug or the photosensitizers are exposed to a specific wavelength of light, it can produce a form of oxygen that will kill the nearby cells. So basically, this therapy is used in killing the cancer cells. So now this is the methodology part for this paper. So the first part is to design the principles for peptide sequence and supramolecular peptide. So the peptide sequence is the G7-CC-ERDGS. So G refers to glycine, C refers to cysteine, E refers to glutamic acid, R is arginine, D and S are aspartic acid and serine. So the first design, which is the multiple hydrogen bonding, which is close to the hydrophobic core. This design used 7 glycine moieties to form the strong multiple hydrogen bonds. And the second part is the employment of cysteine unit that is easily oxidized to form the disulfide cross-linkers by exposing to the air. Because by using this, it can form a crucial part in these linkers in order to maintain the functionalities of the peptide as well as the self-assembly properties. And the last part is the ERDGS, which is the heading of this sequence, which promote the cellular internalization of the self-assembled nanoparticles with the cancer cells. Now, from the figure, we can see at the end of the part of the peptide sequence, it has the bromoalkyl group, or at the end terminal. This bromoalkyl group acts as the hydrophobic component during the self-assembly process. And most important part is that this bromine atom can easily be substituted with 4-methylpyridine to generate a cationic gas in the form of pillar 5 or P5. P5 is a water-soluble pillar that can act as hydrophilic at room temperature and can become hydrophobic as the temperature rises. So, the utilization of P5 in this study is good because it has a thermoresponsive property and a strong binding affinity. So, with these two best properties of P5, it can be used to attach to the peptide sequence in order to control the self-assembly behavior without to go through with tedious covalent synthesis. So the molecular Lego part in this study is where the utilization of both P5 and PYP, and these two can aggregate to assemblies including the sheets and the nanoparticle control. Now, the second method is to study the host gas composition and thermoresponsiveness, and the third part is the fabrication of a supramolecular peptide and its programmable self-assembly, and the fourth part of this method is the encapsulation of photosensitizer and singlet oxygen generation. So from the figure, we can see the first step, which is the study of the host gas composition and thermoresponsiveness, is at the top part of this figure, which is we can see the gas used in this study is G1, and then for the third method, which is the fabrication of a supramolecular peptide and its programmable self-assembly, is when the P5 is combined with the peptide sequence, which can form the supramolecular peptide self-assembly. When the P5 is in the hydrophilic properties, it can form the sheet-like aggregates, while when it is in the hydrophobic state, it will form the nanoparticles. The nanoparticles will then combine with the TPP or the programmable peptide to form a combination in order to encapsulate in the cells, which is performed in vitro. So, this combination structure will then bind with integrin in the cells before going into the targeted cells. The sole reason or the benefits of this study is to enhance the efficiency of photodynamic therapy. In the paper, it has stated that the resultant nanoparticles with targeting ligands on the surfaces are suitable carriers for the hydrophobic properties of the photosensitizer in order to enhance the PDT efficiency. So for the future prospect part, although this PDT treatment has been long established, but it has not come to clear recognition because one of the reasons is due to the low selectivity of the photosensitizer. So, to improve the photosensitizer, it can use the conjugation of the moieties such as the antibodies, polymers, or peptide scaffolds. Other than that, we can go for the characterization of the process of the self-assembly in much more rigorous manner. Rather than using the proof of concept experiments, we can go for the quantitative assessment, as well as we can go for the understanding the roles of thermal fluctuations and kinetic factors, which is important in the formation of the structures. So for this part, I will put all the link to the papers that I use for reference in the description box below. That's all from me. Thank you for watching.